Hello, everyone, and thank you for attending my virtual poster session. My name is Kiersten Campbell, and I'm currently a postbac fellow at the Bioinformatics and Scientific Programming Core of the National Institute of Child Health and Human Development at the National Institutes of Health. In collaboration with Dr. Ryan Dale, this project explores the power of interactive learning for enhancing user understanding of the DESeq2 model. To give some background on the motivations for this project, DESeq2 is a widely used software package to analyze count data, notably used in differential expression analysis for RNA-seq data. This package emerged as a powerful tool for addressing key challenges in differential expression analysis, such as low power due to small replicate number. However, due to the complexity of the statistical model used by DESeq2, users may have difficulty understanding the nuanced behavior of DESeq2 depending on their statistical background. While there are many existing tutorials and vignettes that describe how DESeq2 works, these educational resources are not constructed to be interactive. We aim to create an interactive teaching tool which would allow users to drive their own learning process as they explore various parameters relevant to differential expression analysis, and therefore foster their own intuition on DESeq2's approach. By allowing users to dig under the hood of DESeq2 and explore crucial details of its statistical workflow, we also aim to improve users' confidence in the differential expression results produced by DESeq2. The DESeq2 interactive teaching tool is implemented as a Shiny app, which allowed us to create a polished app with reduced programming overhead. Within the app, users are able to manipulate various input modules, such as a number slider. In the backend, these user specifications are tracked as reactive values, which are essentially variables that update in real time according to the user specifications. These reactive values, in turn, are used to render other components of the app, such as a data table. In the context of the DESeq2 interactive teaching tool, these reactive outputs allow users to manipulate parameters relevant to differential expression analysis and see the corresponding impact of those parameters on the concept of interest. Let's now take a look at a demo of the app itself. You can see that the teaching tool is broken up into key steps of the DESeq2 model and are structured as different pages. Users are able to self-pace their learning by exploring each step in the workflow in as much or as little detail as they feel needed. For example, a motivated user may spend considerable time on each page and conduct thought experiments manipulating different combinations of the parameters, or another user may find it sufficient to just read the text explanations on each page. One of the first concepts addressed in the tool is the structure of the input data provided to DESeq2. This text here will give a brief overview of how sequenced RNA-seq reads are transformed into gene counts by aligning to a reference genome. The subsequent text then begins to explain the structure of the input dataset, which consists of a count matrix with rows representing individual genes and columns representing individual samples. If users want to see an example of a count matrix, they can check and uncheck this checkbox to show a generated dataset, and then they can manipulate different parameters of this dataset using these sliders. So for example, they can increase the number of samples and see how that changes the count matrix, decrease the number of samples, and hopefully this will reinforce, again, that the columns in our count matrix represent the number of samples that we have in our dataset. You'll notice that each of the columns is colored, and they're colored according to a sample group specification. By default, our tool will split the input number of samples into two even sample groups. And then the subsequent text following the explanation of the count matrix structure will explain that the aim of differential expression analysis seeks to look within each gene row and assess how the variation within a sample or within a set of the same colored columns, how these gene counts vary within a sample group versus across sample groups, so by comparing these different colored columns. And again, users can manipulate how different variance parameters affect the gene counts and take a look at how that affects the resulting generated counts. Another key component of the DESeq2 model is the negative binomial model, which is used to model gene counts. This text explains how a negative binomial is parameterized by a mean and a dispersion. But what if the dispersion parameter is unfamiliar to the user? Well, they can use this slider to explore how dispersion will impact our negative binomial distribution. So for example, if we increase the number of samples to get a better look, to get a higher number of observations for this distribution, they can increase dispersion and see how now our range of values increases. And so that'll show us that we now have observed recounts that are much further from the mean. And if we decrease dispersion, we can see now we've got a more even distribution around the mean. And so users, after several trials, can begin to understand that dispersion will describe how much gene counts vary around the mean. Looking towards future directions for the project, we'd like to continue refining the app and ensuring that all of our interactive plots are really addressing our user learning goals. We'd also like to improve the underlying model complexity in our sample data sets. For example, users may encounter situations where there are different replicate numbers across sample groups. There are more than just 2 sample groups. And we'd like the tool to be able to reflect those real-world RNA-seq data sets. And additionally, we'd like to start testing the tool with users who will help us note different areas of improvement for future students and future releases. Thank you again for taking the time to listen to my virtual poster session. If you'd like, you can explore the project further using the poster PDF, which is linked in this session, and I've also included my contact information below. The tool will be available soon on GitHub, and again, thank you.